So thank you, Lori, and thanks, Jeff, and everyone for having me here today. It's nice to be back here again. So I'm going to talk about, so you're probably wondering why, if Lori just said my talk was going to be about MODY, and I said that my talk was going to be about precision medicine and diabetes mellitus, what's that all about? Well, because really I see the work that we're doing in identifying and diagnosing and treating monogenic diabetes as kind of the beginning of a bigger picture of how can we use genetics and other tools to improve how we diagnose and treat diabetes. And we sort of, these are kind of the simplest cases, so I think in order for that to happen on a widespread level, we need to look at the kind of cases that are in some way simpler. And my interest in this came about because, as Lori mentioned, I am a genetic counselor, and when I was studying to be a genetic counselor in the 90s, I was learning, and I was very intrigued that there were certain forms of diabetes that were monogenic. Then by chance, I happened to, after working briefly in the field, happened to move more into research and started working with a research group studying type 2 diabetes. And pretty soon I was involved in analyzing data, and pretty soon I was involved in writing grants. And I noticed that we were writing a lot of grants where we were saying that the purpose of what we were doing was to identify genetic components in type 2 diabetes, because by doing that, we could improve the way that we diagnosed, we predicted, and we treated diabetes. But at the same time, what I was seeing happening was a bunch of papers coming out of the UK, where there was a group at Exeter that was diagnosing people with MODY and dramatically changing their treatment because of their genetic diagnosis. So people who had been on insulin sometimes for 10 years or more were finding out that they did not in fact have type 1 diabetes, but because they tested positive for a mutation in a certain gene, were able to go off of insulin and go on a particular oral medication. And so that was just very intriguing to me, because I came into genetics from a kind of practical patient care kind of point of view. And so as I'm following this, I'm seeing that very little of this is happening in the United States. And so that's kind of the context in which I got into this area. I'm gonna start this talk by, just because we're talking about a specific form of diabetes, but we're also talking about diabetes, I'm just gonna do a brief kind of review of what we mean when we're talking about diabetes. And mostly when you talk about diabetes, you're talking about diabetes mellitus. And so just reminder, I think most of us are familiar with this, but just as a reminder, it's diabetes mellitus is a group of metabolic diseases, and they're defined by having hyperglycemia. And there are a lot of different causes. There's a lot of different ways you can cause hyperglycemia. So you can have problems with insulin secretion, you can have problems with how the body responds to insulin, or some combination. And the consequences that you get damage, the hyperglycemia damages organs, especially the eyes, kidneys, and nerves in the kind of macrovascular complications. And then people with diabetes are also prone to cardiovascular disease due to the relationship between the metabolic system and cardiovascular disease. And of course the prevalence is very high. So the most recent estimates have about, just in the United States alone, about over 29 million people. And so as I said, diabetes is a group of diseases. It's classified into see if I'm doing this right, into
the button on
the button.
There you go.
Yeah, so it's classified into sort of 4 categories, not 4 types, but kind of 4 different categories. And so type 1 diabetes is the type of diabetes that also has been in the past called juvenile diabetes, or insulin-dependent diabetes, and it usually affects children. It's usually due to an autoimmune destruction of the pancreatic beta cells. And I'm using a lot of usuallys, because the reason we now call it type 1, it can happen in adults, it can have different presentations in adults and in children, and so it's called type 1. Type 2 diabetes is really kind of a diagnosis of exclusion. It's kind of it's diabetes that doesn't fit into one of these other categories. It's kind of it's where you have diabetes, but it's not due to the destruction of the beta cells. There's this third category of other specific types. That's where there's a known etiology. And so some cases that were originally thought of as type 1 have now moved over here. So those include both genetic forms of diabetes and other forms like steroid-induced diabetes, transplant-induced diabetes, injury-induced, certain chemical exposures. And then there's also another category, which is gestational diabetes, which is not included in this pie chart because if it's true gestational diabetes, it's just affecting pregnancy, although it's a risk factor for either developing or having uncovered future diabetes or having uncovered pre-existing diabetes. And just, this reminder that there's just the message here really is that there's just lots of ways that you can get diabetes because it's complicated. Trying to maintain glucose homeostasis is central to human existence because it's where we get our energy. And so, you're supposed to have your pancreatic beta cells are in response to glucose being in the blood from either diet or endogenously to produce insulin. The insulin stimulates the formation of a larger molecule called glycogen, but then when the blood glucose when the blood glucose rises, you trigger that. There's some other pathways that are less predominant, but also there are counter-regulatory hormones produced in other cells called the alpha cells, like most notably glucagon, that also help with that regulation under conditions such as fasting. So where the low blood glucose, not only sometimes does the glucose go up with low blood glucose, but during prolonged fasting, you have hormones like glucagon that stimulate the release of glucose from the liver. And this is just to remind ourselves, I was showing you glucose homeostasis, and so that's primarily involving the liver and the pancreas, but then insulin resistance has to do with you might be making insulin fine, but there's a problem with how the organs, mainly the fat and the muscle, are taking up the glucose in response to insulin. And so we show these pictures of muscle and fat cells here. And so that being said, I want to just sort of start my talk about the genetics with a case. So this was a little girl, a case reported in the summer of 2006, who was a suburban Chicago kid who at 6 years old had been dealing with diabetes almost her whole life since she was 1 month old. She had type 1 diabetes, but in August of 2006, she checked into the Clinical Research Center at the University of Chicago, and 4 days later checked out starting to make her own insulin. And I once talked about this case after a woman talked about ways to help islet cell transplants work. She did not have an islet cell transplant. It was her own islets, and by the time she was in first grade, she did no longer needed her insulin pump. And so, how is this possible? We'll get into that in a minute. But before doing that, I just want to give kind of an overview of what we've known about the genetics of diabetes. And so, I'm talking about very specific genetic forms of diabetes, but really just about all diabetes, almost all diabetes has a genetic component, a strong genetic component. And we've known that even before we were able to study genes, because we saw strong familial aggregation patterns where you'd see, the rate was, just relatively low in the general population, but in monozygotic twins, if your twin had it, you were very likely to have it, and if you had a sibling, it was still elevated, but not as elevated. Of course, this also showed us that there was more to type 1 diabetes than genetics, because it's not 100% concordant in monozygotic or identical twins, as you'd expect if it were completely genetic. And so, so then the question is, are there genes? And so this suggests yes. And in fact, we now know that there are genes involved in the development of type 1 diabetes. And typically, in the sort of way we're usually taught genetics in school, as you guys, I'm sure are familiar, is we're used to kind of thinking about, is there kind kind of a gene that causes, or a gene where a mutation causes a disease. And of course, for complex diseases, we get, it gets more interesting than that, because there's going to be some kind of environmental factors that are interacting with the genes. And so then it becomes, well, what are the genes? And it's been known for a long time that a lot of the genetic susceptibility for type 1 diabetes comes from certain variants in the human leukocyte antigen region, the immune system. What?
I don't know if the microphone is there, but maybe you could ask people to mute themselves.
Oh, yes. Could people mute their phones for now? We'll have questions, I think, at the end, at the last 15 minutes or so. Thank you. And it's also been found that there's a polymorphic area near the insulin gene that accounts for some of the variability. And then there's kind of just a lot of different genes. And, and then the environmental factors are probably less well understood, but there's been some that are looked at and some that seem clear, certain viruses, toxins. There's been a lot of work looking at whether early exposure to cow's milk causes some of those autoimmune problems. And then just a lot of mystery. And this is just this is 2000. This is kind of what the kind of actual genetic picture is thought to look like. And so what you can see here in this graph is that at the bottom you have all the different genes or genes near regions that have been found in various ways. Some of the older studies, and then some of the newer genome-wide association studies. Here you have the odds ratios, the effect size of these variants that have been found, and you can see that HLA it's off the scale, and some of it's shown as having close to a 7-fold. There's actually some alleles that even go up to 11-fold, and then insulin has an effect. But most of the variants that have been found, like in a lot of complex diseases, just raise the risk by a little bit. And there hasn't really yet been a use found for genetic testing in type 1 diabetes on a clinical level. There are a lot of studies where they are genotyping people, sometimes even babies, mostly for the HLA region, because they're trying to track them and see if you can see what exposures are going to trigger the susceptible people. There have been some studies in those populations because they are having testing of how the families kind of deal with the testing psychosocially. Type 2 diabetes, you have kind of a similar story. In some ways, type 2 diabetes, though it's mostly adult onset, it's a different disease, and it turns out to maybe have a stronger genetic component, so a little bit higher likelihood if one twin has it than another. Of course, that gets complicated because twins are also more likely to share the tendency to be overweight, which is a big risk factor for type 2 diabetes. But also type 2 diabetes, just from early familial aggregation studies, had strong evidence for having a genetic component. And the way I think of what the genetics look like in type 2 diabetes is this. So similar to what I showed before with type 1, the question people always ask is type 2 diabetes really a genetic disease? Are there genes that are involved in the development of type 2 diabetes? And my answer is always yes. That's why we're studying it. It is also true that some people will say, well, we know what causes diabetes, type 2 diabetes, it's lifestyle. And so that's kind of what we think of as obesity, as kind of a lifestyle or environmental risk factor. So the way that obesity is often characterized is it's just kind of an energy imbalance. So you have too much energy coming in, not enough going out, and so there is there is a behavioral component, but there is also there's also a genetic component, and this was known also from familial aggregation and other studies, and now genes have been specifically found that predispose to obesity, and some of those end up predisposing to diabetes, but through their connection with obesity. And then, of course, we don't know exactly how all this works, but we are finding lots of evidence that some of the genetic component of obesity has to do with how it affects these behaviors. So these behaviors could be genetically affected. Sometimes people are skeptical about this, but it actually makes perfect sense, because if we all know there's people who have different preferences for physical activity. Some people might try, once they try it, they might realize they like it. Some people try it and they never get those endorphin rushes that my sister gets, from running marathons. And so, and same thing, some people may prefer the taste of energy-dense food. Some people may, enjoy food less. So that's important. But really, diabetes itself is not really an endpoint. Diabetes causes complications that we talked about. So really, what you end up wanting to know is, what causes the complications, and some of that has to do with how well managed the diabetes is. And some of how well managed the diabetes is, not just for medication, but all of these other factors, including genes. There are studies looking at genetic factors in complications in people who both have diabetes, but not everybody gets kidney disease. But then the other important thing to remember, and I think is just good to remember for all complex diseases, is that this is not a one-way street, and all these things are then affecting the gene expression. So that's sort of how it happens. So that's really hard to study, but even though it's really hard to study, people have studied it anyway. Lots of people have spent lots of time, recruited lots of people, and they found a lot of genes. There's probably like over 100 well-validated genes or genomic regions. These were some of the beginning ones, but now there's over 100 well-validated genes or genetic regions involved in type 2 diabetes. But they still are explaining probably 15% or so of the genetic variants, and that has to do with all kinds of speculative reasons having to do with rare variants, and people even starting to look at rare variants. We're not seeing it, at least at a population level, as quickly as we'd like to, but there is starting to be lots of sequencing done. So lots of great research coming out of type 2 diabetes genetics, helping to learn about new mechanisms. One of the highlights was finding that this gene right here, SLC30A8, which encodes a zinc transporter. It turns out people who have a loss of function mutation in that seem to be protected from diabetes. So that kind of thing is huge for developing approaches for treating diabetes, even when people who don't have the genetic mutation. So having said that, in most cases of diabetes, we're not quite there with precision medicine, or at least is sometimes the way sometimes we think about it. But so what does that mean? So what happened with Lily? Well, it turns out that Lily did not have. Well, she didn't have type 2, we knew that, but we thought she had type 1. She actually didn't have type 1 diabetes. Lily had a mutation in a potassium channel that sits on the surface of the pancreatic beta cell, and it responds to ATP. And this channel is supposed to close, and when that channel closes, the membrane gets depolarized, which allows calcium ions to come in and ultimately for insulin granules to be released. And these mutations that there are, some of them affect the formation, the conformation of the channel or their ability to bind to ATP, and they don't, and so they just stay open. They can't close, and so that's what results in a very severe-looking type 1 diabetes phenotype. But this is the receptor for sulfonylureas, which used to be the first-line treatment for type 2 diabetes, and are still used somewhat, but they're not as prioritized because of safety and so forth. And it turns out that these kids, if you give them high doses of sulfonylureas, in most of them you can force that channel closed, and then it can function normally, and they end up having mostly fantastic glucose control, and of course you're treating the underlying problem. And so this is just exciting in itself. If just to have that happen to one patient, it turns out it is very rare. Neonatal diabetes only happens in like 1 in 100,000 people, so that's rare. But it turns out that it's really important to do genetic testing on all those kids, because over 80% have a single gene cause of their diabetes, and about half of that is due to a mutation in one of the genes that encodes the channel. And so many of them will benefit from switching treatments, but it's not really something you can do lightly. They have to check into the hospital. They have to have certain injections to change their body's response. And so this would just in itself be, quite a miracle. But what also has struck me about this case is that the way that she got diagnosed is that Andrew Hattersley, who leads a big monogenic diabetes group at Exeter, had given a talk at the University of Chicago and had about this, not about Lily, but it was before Lily, about all these cases that were able to convert over to get off insulin. And Lou Phillipson, who's the director of the Diabetes Center, then went to give a talk just generally about the state of the science on type 1 diabetes to a Juvenile Diabetes Research Foundation local chapter, and he mentioned this in passing because it was a cool story, not this, but the previous findings, but he said, but this is, it's so rare, like I've never seen a case that young, and none of my colleagues have, it's just, so rare. Well, it turned out that for one of the board members, it was not so rare because one of the board members was Lily's father, and approached him, and that's how Lily ended up getting connected with, sorry, I'm waving around the pointer in the audience, that's how Lily ended up getting connected with the testing and then ultimately the treatment change. And so I was interested before this and became very interested in, how do we systematically identify these kids so that you don't have to have that specific connection to have this happen. And as I said, neonatal diabetes is very rare, and but what's not as rare are other forms of monogenic diabetes, maybe a little bit less dramatic sometimes, but also have great possibility for precision medicine based on the genetics. And so these have been, for the past 50 years or so, have been called maturity onset diabetes of the young. And that's because it was observed by some really careful people that there were some kids who in the 60s, 70s, and they had diabetes, but their diabetes was more like an adult diabetes in that they could produce insulin. So they had impaired but not absent insulin secretion. So they sort of arbitrarily said, well, it's happening early, like as early as before 25. And they also noticed autosomal dominant inheritance, meaning they were seeing it go down through 2 or 3 generations, which is sometimes hard. It's hard to distinguish sometimes because of diabetes, other types of diabetes cluster in families, but this was, would be more consistent. And then the other thing was obesity was not required. So I say required in italics because it became sort of known as kind of a lean diabetes phenotype. The problem is it's completely compatible with obesity. So if you're in a population and a time and a place where rates of overweight and obesity are high, then you're going to see people who have monogenic diabetes, have maturity onset diabetes of the young and are overweight or obese. There's a lot of different single gene forms. There's been 14 genes that have been called MODY genes, mostly involving transcription factors in producing insulin or developing the beta in other cells. There's one that kind of has a phenotype that stands out, which is glucokinase, or known as the glucose sensor. It's the enzyme that phosphorylates glucose as it comes into the beta cell and sets off the insulin production cascade. And then there are others that are not identified, and we're starting to get better at identifying it. We may not be using this term as much, and in fact, some of the experts want to abolish the term. And just also wanted to mention that what's really crucial here is that it can, if you're not looking for it, it can look like type 2 diabetes or type 1 diabetes or gestational diabetes. So there is some interest in getting rid of this term for a few reasons. One is that we're seeing type 2 diabetes in children that's not necessarily monogenic, but that's more due to increases in overweight and obesity in children. And also that we know more about the etiology. So it's been suggested to call it transcription factor or glucokinase diabetes. But as I think Lori demonstrated, we can't just stop using the term MODY altogether, because what I found is that when people do know about this, like clinicians, when they know that there's such a thing as monogenic diabetes, they know the term MODY, and it's very catchy. It's an easy to say. So I kind of use both, but then I refer a lot to monogenic diabetes. And just this is from a classic paper from 2001, and what this is showing, basically, at the time there were 6 MODY types. 5 of them were caused by mutations in transcription factors in the nucleus of the beta cell, and then you had this MODY 2 that's caused by glucokinase mutations, where you have a defect in the phosphorylation of glucose. And so the phenotype of this, the disease that, or the condition that people who have reduced activity of glucokinase in their beta cells have is that they even starting from infancy, they have slightly elevated fasting glucose that may reach into the prediabetes or diabetes range. You often won't know about it because they don't have any symptoms, but when they are but then they have a pretty much normal response to a glucose load, and that's because when there is glucose around, the glucokinase is able, the small amounts are able to function better when glucose is around, and they're actually able to process glucose coming in. But they actually have an elevated fasting glucose, and often only detected during pregnancy, and because that's when women are screened, young women are screened for hyperglycemia, whereas that age they're not necessarily screened to begin with. But getting back to the transcription factor types, the most commonly known transcription factor type of diabetes is, it's HNF1 alpha diabetes, also known as MODY3, and so it's been shown that, similar to Lilly, they can also use sulfonylureas. In this case, very low doses of sulfonylureas because they're very sensitive to it. And so there have been trials done where they've shown that most if you can, the ones that agree to transfer from insulin to sulfonylureas, and are able to, that most of them have, not all, but most have been able to remain on sulfonylureas and have decent glucose control as defined by HbA1c, and only one where it deteriorated, but they're at least at the target HbA1c of 7.5, and some of them actually improve glucose control. And so you can see that you have that opportunity for intervention. And this also has profound effects on their lifestyles. They talk about changing, not seeing themselves as diabetic anymore because they're going from using insulin to using a pill. Yes?
Any idea how that works? The sulfonylureas and transcription factors?
There is. I think it's not completely understood, but I think it's the idea that because I don't know. I'm going to actually defer to Mark on this one, so we'll make sure we'll get it correct.
Yeah, it isn't absolutely clear, and it's probably just the continuous increase in insulin secretion that overcomes the diminished insulin secretory response from those beta cells. So, and once you've got that started, it's why I'm a big fan of trying to find this as early as possible, because beta cells don't like hyperglycemia, even a little bit of it. And so the earlier you diagnose, the better your chances of maintaining reasonably normal insulin secretory activity. But I think that's probably all it is, that you're just working like a sponge and increasing insulin secretion. But they're extraordinarily sensitive to it. Metformin doesn't really work very well in these students.
Right, which makes sense because metformin is treating insulin sensitivity, and unless they have an accompanying insulin sensitivity defect, then it's not going to. So a lean person with MODY is probably not going to respond to metformin. So then I also mentioned the MODY 2 cases, and so those cases, because the fasting glucose is just always elevated, it turns out that they don't. In the MODY 2 or glucokinase cases, they don't suffer from high rates of complications, at least the studies that have been done so far. So it actually suggests, and it's actually very hard to get their fasting glucose to the normal range. And so it suggests that when that's discovered, you actually can take them off treatment and reduce follow-up. But so given all this, I think this makes a really good case for finding all these patients, and Dr. Feinglass agrees. But anecdotally, it just seemed like this wasn't happening. And so then finally there was some data, research data to support this that came out in 2013 from the SEARCH study, where the SEARCH cases this was a multi-center study to look at the etiology of diabetes in a large population, and just all of it, not just MODY, but this was kind of a sub-study of it where they took the SEARCH cases that had that had these two markers measured. So these are markers, these are biomarkers that can be used to partially distinguish between type 1 and type 2 diabetes and other types of diabetes. So diabetes autoantibodies, so people with type 1 diabetes tend to have circulating autoantibodies that are a marker of the autoimmune destruction. And fasting C-peptide is a measure of a component of the proinsulin molecule that gets cleaved when insulin is secreted, and so it can differentiate between just insulin, between endogenous and exogenous insulin. So, so basically, where they took the kids that did not have antibodies and had signs of endogenous insulin production, and they tested the ones that they could for MODY. And by that testing for MODY, I meant they tested for they sequenced 3 different genes that account for roughly 85% of cases. So those are the HNF1 and HNF4 alpha transcription factors and glucokinase. And so And so what they found was 47 of those cases had a mutation that they considered to be a MODY mutation. But only 3 of those 47 had a previous diagnosis of MODY. Here's what their diagnoses otherwise looked like. So, so you can see that about half of them had a previous diagnosis of type 2, which sounds high for pediatric, but remember these were selected not to have the markers of type 1. Nevertheless, a lot of them had a clinical diagnosis of type 1, and as I said, a few had a diagnosis of MODY, and then some kind of assorted other things. But the other important thing about what they found is the ethnic distribution pretty much reflected the people they tested, and so it was found across ethnic groups. The reason this is important is because the American Diabetes Association, up until this year, they used to not have any recommendations about testing. Then when they had testing, they would say, who should we test? Because again, you can't test everybody. So they would say, well, test children who are not from a high risk, who are not considered to be at high risk for type 2. So then you're more likely to see MODY in a kid that's not high risk for type 2. Well, one of the risk factors for type 2 is being from some of these other non-Caucasian ethnic groups. So yes, you would increase your hit rate, but it could be a serious problem regarding health disparities. And so I've noticed they've greatly revised their guidelines this year, January 2017, and they actually no longer mention ethnicity as a factor that might make type 2 more likely and then less likely to test. But then the really most important thing is the treatment. So this shows the treatment they were on by the gene that had the mutation. And circled are the treatment that's indicated by that gene. So the HNFs are generally best treated with sulfonylureas. Very few of them were. And then only about half of the glucokinases were on what's considered the quote-unquote right treatment, which is not any treatment. And there were a lot on insulin and metformin. And you can see a lot of kids on metformin, and that's going to reflect the fact that they had a diagnosis of type 2, and so that's what they were treating with. So why is this? Why is there this disparity between what seems to make sense, like that we should be detecting it, and the fact that we're only detecting like 6% of cases? So over the years, from literature and from talking to people, I've kind of come up with an evolving list of possible challenges. So the most sort of central is just the lack of awareness. If you're going to look for it, you have to know that it exists. But even if you're aware of it, it's still tough because of this clinical overlap. I mean, there are things that distinguish them, but there are also characteristics that the types have in common. So, what do you do to pick these kids out? But even so, I've also encountered, and I've had people express that they've gotten this from a provider, like that rare means never. And this isn't really unique to diabetes. I think we're all, I'm sorry, I'm preaching to the choir here, we're all genetics, but, and so I've had people say, well, my doctor said I couldn't have it because it's so rare. And then said, well, I'm in my 20s and I'm lean. And said, oh, but some people, there is a rare number, there are some people who have type 2 diabetes in their 20s who are lean. Probably many of them don't, but that's sort of what happens. And so the way I kind of think about this is, and I forgot to check if we have audio, but the idea that when you hear hoofbeats, the whole thing about when you hear hoofbeats, think of horses rather than zebras. And I think this is something where, and I think it's funny because in genetics, in clinical genetics, I think people are sort of taught the opposite. You're kind of looking for those rare cases, but I think sometimes it's it's sort of like this is the most likely explanation, but just the importance of always considering all the possibilities on the differential, I think, is sometimes tough to do. The idea that it's life-changing versus life-saving, so some kind of medical breakthroughs, like, Lily had a tough time. She had hypoglycemic seizures. I mean, but still, she had a treatment that most of the time it was generally controlling her diabetes. But for some people, they might even have an easier time, but this could make it even better. So the idea that they already have a diagnosis, they already have a treatment, so getting people on board with, having a test to see if they have a different diagnosis that might respond even better to a treatment. What has been, and this is evolving, is the expense and complexity of testing, and the expense has somewhat been driven by sort of commercial models, but also just that it has been complex because most of the mutations in these diseases are rare, and so you have to sequence a gene, sometimes several genes, to find it. So that's been an issue. Resource prioritization. I mean, this is from starting to talk to people in other countries and other places where, some of the value of this is like you have this person who thinks they're pre-diabetic and they think they're going to get diabetes, and you can reassure them that they're not, and call it like the worried well, and people say, well, here we don't have the resources. We only have the resources to take care of the sick people, not the worried well. So that comes into it. There has been limited professional society guidance. As I said, that's starting to change from some of the efforts that we, and I think in part that from we and others are making. And and then just related to all this, just the time allotted to visits is kind of like another thing to discuss, and diabetes care is very complex. Although, if the etiology were more correctly classified, it may reduce that complexity somewhat. So what we're doing as part of our IGNITE project is called the Personalized Diabetes Medicine Program, and it's designed to try to attack all these barriers at the same time. And so we've designed this approach where first we want to identify the patients. We've been doing paper questionnaires. We're starting to transition this more into online questionnaires at some of our sites, and also putting standardized questions into an EPIC dot phrase for the clinicians, but basically asking some very simple questions where the answers are not necessarily in the chart, because this is the third or fourth provider. It may not say what their age of diagnosis is, but knowing these things helps us with distinguishing the types. Then we also get their C-peptide and antibodies. And if based on all this they still look suspicious, then this is all kind of screening. Then we invite them into the study, and then we get more information from them. And if based on their family and medical history, there's a reasonable chance that based on what's currently published, they might have monogenic diabetes. Then we sequence. We have our own in-house sequencing platform for sequencing the coding regions of 40 genes. Right now, we're doing that on a research basis. We're probably at some point going to move into a clinical panel. Right now, it's a research basis, but if we classify those variants, if we find one that we can classify based on ACMG criteria, American College of Medical Genetics criteria, which are very complex and very stringent, as a reportable variant, then we tell the patient, we put all the information in their health record, we communicate it to the provider, and we also are making genetic counseling testing available to family members where they only have to test for that one variant. If we found one of unknown significance, then we're looking at that more closely. We're doing more family studies that can help us find the evidence of whether it's important. This is just a quick show of the genes that we're currently looking at and why we're looking at them. And this is the one so in doing this and applying the standards and guidelines for classifying variants, we came up with and published. Oh, okay. This is why I have a plan B, which is a screenshot. This is a screenshot of a tool we developed because basically there's these two organizations developed this system trying to, because you find a variant in a patient, it's never been seen before, it's only been seen in a few. How do you decide if that is just normal variation or if that's causing disease? So the answer is that you look at all different evidence, and they have this way of looking at all this evidence. We came up with this kind of graphical user interface so that we can enter the evidence in, like what functional studies were done, and and how many families has it been seen in, and then at the bottom it calculates for us whether it's pathogenic or likely pathogenic or unknown, and then we can use that to develop a report for the patient. And then our program, it's a part of the goal of Ignite is to disseminate across especially to non-academic centers. So we're also working with our VA, we're working with an integrated health system, and we're working with a local large private practice, large endocrinology private practice. We're also looking at we trying to get more out to other local clinics and talking directly to consumers. We'd like to we're really trying to develop some clinical decision support that would be exportable, so that no matter so that in addition to education, it becomes more and more automatic to be able to ask these questions, and then starting to work with professional societies. This is if you've seen anything about Ignite, this is the whole map of Ignite. So you can see it's a network of networks, and so as Lori said, Ignite is an NIH network to do projects to look at implementing genomics in practice. And so as an Ignite project, this tells you a little bit about the evaluations we're doing, looking at the clinical outcomes and the patient report outcomes, what's working, what's not working, looking more closely at the experiences of people involved. And we're also engaging with the payer community and people who are making decisions about getting these things covered. We've discovered about we've identified about a dozen patients with mutations. I have lots of stories to tell, but one of the stories was a woman named Sophia, who agreed to have her story told in public. So Sophia is a kickboxer, as you can kind of see from the picture. And she had been told in college on a routine screen and in her 20s on a routine screen that her glucose was high. She was sort of admonished to kind of take better care of herself. Like, you got to start exercising more and, eating better. And she said, well, I can't possibly, because I'm in this really intense training program, and I eat I eat a very low carb diet. And so she just kind of knew something was wrong. She ended up in our study, and we found that she does have a glucokinase mutation. So her hyperglycemia is caused by an enzyme defect that can't be corrected. So she, I think, and I just have her to thank, because we were able to, for one day, get monogenic diabetes out to the whole city of Baltimore. And what happened was they came into we had our PR department was working with them, and they came in to talk to us, and they brought a photographer. We're like, what are you going to take pictures of? Oh, I know. You could take pictures of us talking to a patient. That's really interesting. And so actually, this is another one of our patients also had a glucokinase mutation, and she was diagnosed with prediabetes and then type 2 diabetes. She was on Januvia, which is a combination medication for diabetes, and but then two of her kids showed up as who were young adults who were lean, as having incidental hyperglycemia, and then she was tested and found that this was the cause, and she did not have type 2, so she's now off medication. But when the photographer came, Sophia wasn't able to be there because she's a teacher, and so the reporter phone interviewed her, and then unbeknownst to us found out that she was a kickboxer, and then realized so I attribute her for really helping us to get it on the front page, because she gave us an action shot. Just some further directions we're going in. As I said, we're customizing the electronic health record templates. This is a different way of screening. We're looking at more things that providers would know, and going into this tool that the University of Exeter has published. We're looking at telemedicine models. We're realizing that the model for this might be like a few centers across the country that can provide this service remotely, because it's a little bit complex, like what it means to have a mutation. Trying to bring people together, patients and providers together, try to do provider training, develop educational materials, and really do collaboration both nationally and internationally with other people who are concerned with this as well. And the project that we're doing, it takes a lot of just within our own institutions and centers, takes a lot of of work and a lot of people working together. And also, as I acknowledge, as a funding source, this is a woman that I met at a screening of a movie about monogenic diabetes who had been recently diagnosed with MODY 2, and she seeded a fund in our UMB Foundation to look at raising money for doing work and advocacy in this area. So I'd like to thank everyone for listening, and I'd like to take any questions you have.
I have 2 questions. So related to implementation, right? So 1, is there ever going to be like a panel that you could order? Because you already said how complicated it is to get this task done with the sequencing. If we really want to get this out there, there needs to be an easy way to do that. Is it possible that it will ever become a panel, or is it always going to be a complex task to order?
That's a good question. I mean, there are, as I said, we have this panel that we're doing on research basis. There are some clinical panels available, and so there are panels available, and there's clinical testing to order. I think people are still people are still struggling to get it covered, so it's available. But I guess what I see happening, and we're at this crossroads in genomics, where at some point, people are going to start to have their genome available, and it'll be a matter of looking at a genome. But I think for now, what we're trying to do now is we're trying to change the. We're trying to looking at what genes there's. You can see that a lot of it's important to know all of these genes because of the implication for the family. But there's a handful of them that have very specific clinical actionability, and so we're looking at trying to come up with a test code that will cover those genes, and we think that might make it easier for people to get it covered because the indication is very clear and the outcome's very clear. But part of what we need to do is get the outcomes data from our study to show that actually doing that test actually leads to the outcomes that we say it will. So
that's good, though. I think will help you with the sustainability part.
Yes, exactly. Yeah.
And the second question then is along the similar veins: sustainability, genetic counseling. You're using a different model for this, right? I mean, you're using telecounseling, but are you using a traditional pre-visit genetic counseling? This is DNA. This is what the test results might look like. And then
We are, for our patients, they do have, as part of the informed consent process, they talk to the genetic counselor, and the genetic counselor explains to them the tests we're doing. In part, it's really important for our study because we have to explain to them that you don't get a negative result, you get, either no result, or no, you don't get no result. You get a letter that's saying it's inconclusive or result. But the post-test, I think it's important for the post. So yeah, and then they're getting the post-test. Now, in terms of scalability, I hear what you're saying, and I think, so we've grappled with this a little bit. One thing we're doing is, obviously we're trying to educate people about it, but I think what we're realizing is it's not a rare condition, but it still is uncommon. And so a model that might work better, instead of trying to try to expect everybody in the whole world to know exactly what to do, is to have a small number of centers where they can do this sort of more intensive, at least especially the post-test counseling, so they really know what to do with the results. And the other thing we're doing, we're looking at ways to educate the genetic counseling profession, because we still figure there's going to be a handful of people, but we think that probably every genetic counselor, they need to know, and if they do know, they would be equipped to then know what to look for in a family history, and then be able to. So we've actually published a potential, an algorithm that genetic counselors could use, and potentially other providers, but genetic counselors, it kind of goes with kind of how genetic counselors already do things. And then, yeah, so I don't know if that helps answer your question, but it is challenging.
But you don't necessarily think that somebody else besides a genetic counselor could be trained to do perhaps the pre-test part?
Oh no, I think they could. I think there's definitely room for, right now we're sort of looking at, okay, this hasn't really been done. There hasn't really been a model come up with to do this. So we've been focusing on, okay, this is we're trying to develop kind of a gold standard here, and then what parts of it can then we place, and I would imagine that there would be, some of these cases are straightforward. I mean, there's an algorithm that's published that suggests that every pregnant woman who was lean before her pregnancy and has elevated glucose should be tested. And that's the kind of thing where you could work with the OB clinics to do that initial screening. And then if you find a mutation, then it might be either optionally or referred for. So yeah, so I think there's going to be scenarios that can be scaled up like that. But it's sort of the first step is figuring out how you want to do it and what you want to teach. So yeah.
Talk about that tool that incorporates the evidence and the complication that goes into medicines.
Oh yeah, yeah. Let's see. Oh, well, I was just going to pull up the tool first. From current, yeah, there we go, current slide. Yeah. So the calculation itself is actually pretty simple. It's the guidelines that are complicated. So the guidelines there are a number of pieces of evidence, and they're labeled like, it says PVS1. So this is like very strong evidence. This is very strong evidence of pathogenicity, strong evidence of pathogenicity. And the way this works is they list all these different criteria, and each of them has a different definition. Like PVS1 means it's a null variant, either nonsense frame shift, splice, and then PS2 is like de novo in a patient with disease and no family history. So so they define the original paper that came out defines all those. And then what happens though is then there's another table in the paper where it's like, well, you have to have either 1 strong and 3 moderate, or 2 strong and 1 moderate. And so it's sort of a matter of bookkeeping. And so the way we developed this tool is that first the lab director just like made herself like a printed out spreadsheet where she could check off boxes to see how many boxes she checked off. And then I said to my graduate student, Jeff Kleinberger, who's an MD PhD student who's done a huge amount of work behind this, I said, well, this is an Excel. You could just make Excel calculate it. So then I had it so that when you check off the box, if you whatever combination of check, you just have a simple like if-then rule that says, if it has these, then it's these. So it's a pretty simple calculation. So then lab director Linda James, she said, well, hey, this could be useful to lots of people. Let's publish it. So then you don't want to send an Excel sheet with a publication. So we made a website that then the publication could refer to. And so it's just doing the same calculation. So fairly simple. And he has, probably should have brought it, but he has, of course you can get source code from a website easily, but he actually has it in an open source website. People have contacted us and have done some development with it. So, sure. Yeah.
Just sort of thinking about the best way to educate for this, because even though we say this is uncommon, we still are potentially talking about at least hundreds of thousands of people in the U.S.
Right. Thank you for saying that. Yeah.
And my bias that MODY 2 may be a lot more common than we think it is. So the question is, caregivers are so busy, don't take a lot of time. Is the best education to go to the population itself rather than caregivers? Are you going to look at that as your...
I think that's kind of what we're sort of almost seeing that naturally. When we originally conceived this project, it was like going to be in these centers. But we did advertise it, and so we've gotten people self-referrals and provider referrals from all over the country. And it's been kind of fascinating because, so for example, one of our patients saw she didn't see the study advertised. She saw the syndicated version of the Baltimore Sun article, and she said, my whole family is diabetic, but they're also all overweight and obese. And I was determined to not get diabetes, and so 20 years ago, I changed my lifestyle drastically, but my sugar's just always been high, even when I was young and thin. And she said, and now my doctor says my HbA1c has hit the magic 6.5, and so I have to go on metformin. So she became convinced when she saw this that she could have this, and she did turn out to have a glucokinase mutation. And so that was really instructive, because one of the things we've actually struggled with is, we have our criteria, and the criteria really aren't so much to substitute for professional judgment. They're sort of to be something that people could have if they don't have a provider who thinks of this. You know, we don't want only people who are in academic centers who like think about these things. We want anyone. I mean, so we like these to just like get these criteria so that they would just be a standard part of the diabetes workup. But then we also take these professional referrals and that may not fit the criteria very well if we can make a case for it to, you know, and that's to honor and to pursue what, you know, what does it look like when you see millions, thousands of patients and this one looks different. But then we sort of thought, well, if we just open it up to people being able to refer themselves, everybody's going to think they're special. And but that hasn't really been the case. The people who are self-referred, it's like they're really motivated, you know, and there may be other people that aren't thinking of it. So I think that's been like a lesson. So I agree. I mean, the whole idea behind the tool in the waiting room, it's been sort of challenging because, you know, staff turn. It's not always easy to get the questionnaires distributed, but that's sort of been my strong belief is that if you want to teach a provider to do something, the most well-intentioned provider, it's very hard because they have tons of patients to worry about, tons of things to worry about. But the more you can reach individual patients, then they only have they're one person. So I think we should be doing both. And so we're actually, my postdoc recently promoted to faculty, she and another colleague did a website analysis. So they analyzed all the websites on monogenic diabetes for you know, accuracy and reading level. And most of them are like 13th grade or, you know, like college level or higher. So there's not really a lot of good consumer-directed information. And so that's sort of step one. Is doing that analysis, and then the next step is trying to develop better ways of reaching out to the public.
But you know, that's how glucose monitoring started. It wasn't docs saying, oh, you must do this. It was patients coming to the doc saying, this is just out, and I'd like to measure my glucose. I want you to order this for me.
Yeah,
So
I think that,
a lot of things are patient-driven in this case.
Yeah, I agree, and I think, you know, sometimes geneticists have like cringed about some of the patient-driven stuff, you know, but you know, like some of the genetic tests that, you know, that are available, and sometimes they, you know, I imagine other providers do. But yeah, I think it's really important. I think the patient-driven side is really important. I think we need to engage more, have more engagement of patients in kind of thinking about all this. Because every time, I mean, we're doing some qualitative interviews with patients, and the reason we're doing that is because every time I talk to a patient, I've learned some branding thing that that wasn't in the, I felt like I learned a lot from the literature, but then I learned just a lot more from talking to people, so definitely. Yeah.
You mentioned earlier with your survey tool that you guys were starting to integrate that into the health record. Is that a route that you guys are looking at, like creating a clinical decision support tool based on that questionnaire where if so many answers are responded a certain way that you'll get a pop-up and say, hey, you might want to test for MODY or something?
Exactly. That would be our ultimate goal. The ultimate goal would be the way to capture this information from some combination of information from the patient and or provider, and just have a pop-up that says, you should consider testing this person. And you know, like every infant, there should be a pop-up. And actually, one of the payers we talked to had this idea that, you know, maybe it could be at the pharmacy level. Like the pharmacy gets a prescription order for insulin for a patient who's less than 1 year old. They could bounce it back and say, hey, wait a minute, have you tested this person? And so that, you know, that benefits everybody. So we are doing that right now. The level we're at with the resources, you know, we have and opportunities we have is at Geisinger. They actually have this right now. They're doing the questionnaire as a research tool, but it's the patients access it through their portal, and then. So, but that would be the ultimate. That's been kind of my you know, when I've talked to people, can we just do this like right, you know, just like they fill this out and then there's a program inside that calculates it and it comes up. So yeah, that's the goal. Okay. Other questions? All right. Thank you very much. Thank you.
This is going to go on the Ignite Youth and something else, so you'll see.
I have
YouTube worthy. I'm sure you'll get thousands of hits.
No, I just lost. It's just once.
Yeah, I'm
just secreting waste.
yeah,